On this note, a very warm welcome, a very warm good evening, and a hearty welcome to all for the third expert talk of the International Lecture Series titled Alternative Light Sources for Detecting Latent Biological Evidences on Items. Praying is not asking for what you think you want, but asking to be changed in ways you can't imagine. With these thoughts, we will begin this International Expert Lecture with a moment of silent prayer. I welcome Mr. Don Cairo, Coordinator of Department of Forensic Science and faculty members to this session. A cordial welcome to the students of first and third semester MSc Forensic Science to the session. It is with great pleasure and honor that I introduce Dr. Eugenio D'Orio, a distinguished scientist and an eminent figure in the field of forensic science. Dr. D'Orio's remarkable career and extensive expertise has positioned him as a beacon of knowledge and an instrumental force in advancing forensic sciences. Sir is a luminary in the realm of forensic investigation, serving as Director of Bioforensic Research in Italy. Additionally, Sir holds the esteemed position of Professor of Forensic Biology at the University of Naples, Italy, specializing in Department of Forensic Veterinary. Furthermore, Sir's influence extends beyond academia. He is trusted consultant in forensic biology and genetics for the Italian Ministry of Justice. His expert guidance and insight play a vital role in ensuring the accurate application of forensic techniques within the criminal justice system, leading to fair and just outcome. With an academic background rooted in extensive research and a career marked by pioneer contribution, his work ranges from meticulous analysis of body fluids to the forefront of DNA profiling technologies, constantly pushing the boundaries of forensic research. Today, we have the privilege of hearing from Dr. Eugenio D'Orio, a visionary in the world of forensic science. Please join me in extending a warm welcome to Dr. Eugenio D'Orio. Without further ado, I request Sir to address the gathering.
May I go ahead?
Yes, sir, you can start your presentation.
Okay. Thank you so much. Thank you so much, Dr. Tsushita and Professor Don Cairo. I hope I pronounced well your name. I'm sorry for any accent misleading in case. Okay, it was good. Thank you. It is a pleasure for me to have this lecture with your Department of Forensic Science. So first of all, I would like to thank you for the kind invitation and thank you a lot for the very nice presentation of myself to the student. Well, here I prepared some slide for this lecture and now I'm going to share so everyone can just see also my presentation for today. Okay, I'm trying to put in presentation mode. Actually, it's still short. Okay. Is it visible, right?
Yes, sir, it's visible.
Okay, that's great. So today we are going to talk, it's my pleasure to talk a little bit about the alternative light sources, what they are, how they can be used for forensic investigation, and which are the research that actually are going on these biotechnological instrumentations. I'm going to describe a little bit also my iter, but I was very good presented by Dr. Tsushita, so I have not any more to say anything to say more about me. Just I would like to say that I will refer to this presentation about the alternative light source, about how we started to study this field. Personally, I started this kind of research, the research on this field of the forensic biotechnology, when I was a researcher at the University of Copenhagen in Denmark, 2016 and 2017. Well, as you can briefly, very shortly, you can see here, this is the field of the crime scene investigation with a special focus on the biological and DNA evidence. You can see that, of course, we have the crime scene investigation. So we are outside of the lab, collection of the findings, chain of custody. we are outside of the lab. Then we go into the lab, of course. And what we do in the lab, in a forensic biological lab? Two main things. First is the screening of the findings collected by police or forensic practitioner. And then we do, for selected sample, the genetic analysis. Well, there is here a very big point, because for the forensic genetics analysis, well, we have very shared standard international protocols, international standards for our lab, the accreditation, etc. For the DNA analysis. But the same standard, unfortunately, doesn't apply also for the previous action, which is the screening of items. What I mean, for example, I got in a lab this t-shirt. Okay, it's just an item collected at the crime scene. You can see some signal that may be a biological evidence useful for further DNA analysis, or you can't, because the trace is small, or maybe the color of background just covered the trace. So in that case, if we just go with the naked eye to looking for biological evidence, we have a risk to lose important, relevant evidence for our investigation. Of course, the detection, this is why the detection is a critical point of the crime scene investigation and the forensic biological investigation. If we are able to improve the detection of biological stain, of course, we will be able to improve also the DNA analysis, and this will have a very important forensic application in the court of law in front of the judge. But as I highlighted just before, unfortunately, there are not yet international protocols or standards for performing the screening of the items just before of the DNA analysis. So what we did, what I did when I was a researcher in Copenhagen and I started to explore this field, I attempt the first validation of an alternative light source system. And I'm going to now to describe you how I set it up my research there. Just a bit introduction about the alternative light source. They are a tool who permits to improve the detection of biological evidence. Of course, on the market, there are many different types available. They can be just a small one, but also a big one. Some of these light sources can be used only in laboratory because sometimes they're very big, but some of them, they are very similar to the torch. Of course, specialized torch will emit just a specific wavelength. Well, this one can be used both at the crime scene, both in our forensic laboratories. So now let's going to talk about benefits and limits of alternative light sources. About the benefits, they are a tool that permits to get visible some traces that are not visible with the naked eyes observation. Why these evidence sometimes are not visible? Two main reasons. Very small quantity of the biological stain on the given finding, or sometimes it's just the color of background of the items. I made you the example of my t-shirt. This is a brown one. The brown is a color which is sometimes difficult to get some, for example, saliva spot on it, because the color of background can cover some the biological stain in this case. But it's very important to highlight that the alternative light source is a method for investigation which is not destructive. And this is very important for the forensic application because it permits to redo a second or a third or a fourth test in order to make a cross confirmation of the data that we got during our investigation. This is not possible with the DNA analysis because when we are going to extract the DNA from the nucleus of the cells, well, we can't go back. So that kind of analysis will be never repeatable anymore. And these have a very important forensic application. What are the limits of alternative light source? By the way, well, they are not a new generation tool. There was, of course, a major development of genetics technologies, but related to the DNA analysis in the last 20 or 30 years. The same thing, unfortunately, didn't happen also to this kind of biotechnologies. And I have also to highlight that through a screening of alternative light sources, it's possible to find something, maybe a stain that can be useful for DNA analysis. But we are not able to provide for now a certainly identification of the biotissue. What I mean, we can't say this could be blood, this could be semen, this could be saliva, etc. We can just identify possible stain. Maybe there is some noise in the background. Yeah, please, if you can just mute your microphone, thank you. What was the goal of my research? Of course, the goal was to make a validation study and develop protocols for the application of this technology to the forensic investigation, not guidelines anymore. When I was at Copenhagen University, we had the Superlight S04, which was a very relevant instrument that we have available, and it was the instrument that I started to work with. But this was the technical, instruction, and as you can see, this is quite far away from a real protocol of application. No many specification about the detection power, discrimination between different kind of fabrics investigated, nothing. So we had to build everything from zero, unfortunately. Why I say we must have protocols and not guidelines? Well, because guidelines are suggestions, operative suggestions. Okay, it's easy to apply the guidelines, we know, but unfortunately, guidelines can lead to a very different procedures if we compare to protocols. What this will lead? Potentially to very different results. If we analyze this t-shirt with two different methodology, I can get a result, and Dr. Tsushita, for example, could get different results from mine, but the item was the same, and the tools was the same that we used for the screening. How this is possible? Is this scientific? Of course not. This is why we need standardized protocols. So no matter if I am Dr. Eugenio or if I am Dr. Tsushita, and if I am in Italy or if I am in India or whatever else, we will get the same result. This is what the scientific community have to guarantee to the science first and to the justice then. Otherwise, we are just into the subjectivity and into the probabilistics, which will never help the justice in real way. So how I set up my research? I used this kind of materials. I chose. I had to build a reference sample for make my observation, my calculation, the biostatistics, etc. So I used 5 different fabrics for testing, and I chose the ones that are, most common at the crime scene. So I'm talking about jeans, t-shirt of white color, gray color, black color, and a leggings of blue color. And I used also the 4 different biological stains that we can discriminate through our histological test: blood, semen, saliva, and urine. I use methods. Let's talk about the methods now. It was just like a thesis because it was just postgraduate research. Well, this is a piece. This is an example of a piece. Maybe this image is also easier for you to get it and easy for me to explain. I cut pieces of this fabric of standardized size, 15 centimeter for 3 centimeter, and I divided these into 5 sub areas. Why 5 sub areas? Now I'm going to tell you, because when I do research, I always need at least two things, one negative control and one positive control. So first two subarea, and then I used three more subarea for making some, variation of the stain that I was going to observe and study. So how I did use the alternative light source in my availability at my department. We can move lots of things. It was very IT controlled, that stuff. So I choose a power of the tool from 0 to 10 watts, and I set it up in 6.7, just 2/3 of the power of the instrumentation. Choose the wavelength number 4, which correspond to an emission of lights in between 400 and 490 nanometers. Then I choose a color of googles, because we need to wear googles when we do this kind of investigation. I choose the orange. I had also to choose a filter for my camera, because I had to document it, and the camera have to see same thing of me. So if I use an orange filter, the camera must be provided with an orange filter. Then I choose an angle between the worktop, in which was my items and investigation, and the alternative light source, just perpendicular, and the distance approximately 50 centimeters from the item to my light source. And I made testing in 3 steps. I'm going to explain you. First step, it was the easy one, because it was the detection of the single biological stain at time. But we know that in crime scene investigation, it's very often happens that we find a mixture, DNA mixture or mixed evidence, in which there are more than one contributors. It's pretty common that we found this. So I wanted to study also the detection of two stains together. Step 2, two stains in same concentration. Step 3, two biological stains, but with different concentration. Because we know that in DNA profiling, here there is a big difference. We don't know yet if this big difference is also present also at the beginning level, so at the level of the screening. So let's explore. Well, that's the step 1. So detection of a single known stain. As per I told you before, we had the subarea A, in which we put our negative control, and the subarea E, in which we put our positive control. Positive control, of course, it's 50 microliters, because it was a standardized quantity that we were going to add on this fabric, just of the selected stain, blood, saliva, semen. Negative control, we used water. So 50 microliters, but just water, no biological stain, and a dilution, 1/2, 1/4, and 1/8, okay, of the stain. Step 2, what we did? We did the same approach, just we used 2 stain, for example, blood plus semen or blood plus saliva, and we made all the combination. Always same, positive control, negative control, and dilution in the middle. And same thing we did also here in the step 3. Just we unbalanced the mixtures, the concentration of the sample, stain 1 and stain 2. Well, let's see this. Of course, later I will show you the picture from where I recovered this result. I'm going to explain you this. Okay, this is the detection of a single stain on different items. So black t-shirt, gray shirt, jeans, etc. We did the naked eyes observation. So we are up in the slide. Well, plus means I can see anything. Minus means I cannot see anything at all. Well, as per you say, the major part of the biological states was not visible with the naked eyes observation. So this is a proof of how strong is the needed of this technology for this forensic investigation. Then we took all the negative sample and we rescreened with our protocols that we just standardized. And you can see the red plus, actually. Red plus means that these stains turn out visible, but before they were not visible. So this is a sign that this instrumentation is working properly. We did a calculation, of course. We made this study in double, of course, because we need to see if this kind of study was repeatable or not. And this is a mathematical average of the data. Naked eyes observation, we have an efficiency, so our eyes have an efficiency of 15%. So what this means, that if we don't use any kind of technology for evidence detection, we will lose more than 80% of the possible biological states for a criminal investigation. This is why it's strongly needed to use biotechnology for forensic investigation before of the DNA analysis. Or otherwise, we will just provide very few and very short information to the court and to the investigators. According to the protocol number 1, we had 13 positive sample on 20, which means an efficiency of 65%. Well, it was, of course, not so much, but it was the first sign, the first tangible sign that this system is going to work. So, okay, let's go ahead and let's see what it happened with the other steps. Of course, your eyes have to be trained. As you can see on this slide, you can see semen on white shirt, okay, on white t-shirt. Naked eyes observation on the left. We can't see anything at all. We can't. In this case, we are just risking to lose the evidence. But if you look at the slide, at the picture on the right, in which we use the alternative light source, you can see this spot very marked in the positive control, because over there we have the maximum of the stain, which is not so big. 50 microliters is not that big amount of biological stain, but we are able to identify the stain. Same thing happen here for the saliva on jeans. Not possible to see anything with the alternative light source, and in here just this spot. This spot means there is probably some something that you can investigate for DNA analysis. Is this a fluorescent signs that go back? It's a fluorescent sign. Same thing for urine, as you can see, not possible to see in naked eyes observation, but it was very clear the spot in the alternative light source investigation. But it was not possible, for example, to see the blood on this kind of a black t-shirt. Unfortunately, we didn't get any improvement with this instrumentation. We'll discuss about this later. Now let's go to the second step. So the identification of two stains with the same concentration. As per before, we had here all the combination, blood plus saliva plus semen plus urine, etc., for a total of 6 combinations on the 5 selected fabrics. As you can see, we have some plus, so something that is visible. But wait a minute. We have blood plus something. And we have to keep in our mind that the blood is the only biological tissue that has some pigmentation because of the hemoglobin proteins. So in this case, we are seeing something, but it was just related to blood. You have to be careful about this. Then, okay, and lots of negative. We took all the negative and we screened it through our protocols, and we see that many of them, in this case, many of them, data are positive. Let's make biostatistical analysis. We arrived up to 24 sample on 30. It means an average of 80% of efficiency. That's going pretty good, actually, because we will never get probably 100% of efficiency, but we have to do our best for reach the highest value. And it was a sign that this was working. Here, there is, for example, another image that show the combination of saliva and urine on the blue leggings, not possible to see on naked eye, possible very clearly to see with this forensic investigation. Here it was amazing what we saw with the combination of saliva and urine in alternative light source vision, because it was nothing to see in the naked eye vision. And same here also on the brown shirt. Well, now a question. Step 3, shoe investigation detection of two different stains with unbalanced concentration. We wanted, because we know that the DNA analysis in this case is pretty different. We asked ourselves if this difference was present also in the detection step. Well, as per you just saw for the step 2, these data are quite similar. In fact, we arrive around 63.3%, which is not far away. We know that the DNA electropherogram of this analysis can be very different when you have DNA balanced and when your DNA is not balanced. But this difference is not present here. So we were able to prove that there is a big difference in DNA analysis, but the same difference doesn't exist in the step of the biological evidence identification. So just for make this clear for all of you. Of course, this is another example. Also here we have these very relevant signs, and on the genes as well. Well, that's a summary of efficiency. Well, all our graphs go up, but they are not able to improve the 80% of efficiency. So we did a detailed analysis why we are not able to do better. And we realized that, unfortunately, this kind of protocols doesn't work properly on black and dark fabrics. Probably there is something that doesn't permit a correct identification of the biological stains from this kind of fabrics. So what we did, we changed our protocols. So what we did, the power of the tools, it was 6.7, we turned up to the maximum. We didn't change the wavelength, but we do change the filter colors of our Google and for the camera. Now it's yellow color, and we reduced also the distance between the worktop and the light source. Now it was around 30 centimeters. So we changed these parameters, and let's see if something will get better or not. We got, this is from step 1, so detection of a single stain. We took all the negative samples and we rescreened with these new methods. And you can see the result. Almost all of them turned positive. Of course, I had to do lots of, attempt. It was very long pathway because I had to find out the good combination between all these parameters. But at the end, it seems that I did it. In fact, mathematically, we had this kind of increasement from 50% of efficiency to 65 with the protocol number 1 to 95 with the protocol number 2. Well, now it's working very good. Here there is a comparison because I had to prove you that we were not able to see any kind of evidence with the protocol number 1, as you can see on the left, but we were able to identify the stain with the protocol number 2, and you can also appreciate the difference of the filter in this case, because the color of background of the image is different. This is totally linked for the filter application. Same thing here. Same thing also here, in this case. So we had lots of proof about it. So we can do this also for the step 2. So 2 stains in same concentration. I took all the negative, in this case, the black t-shirt they were all negative, and 4 on 6 data aren't positive. Let's see what this means in a biostatistical way. It seems that we are now able to detect with an efficiency rate of more than 93%, which is still good. Here, some more picture that show that was not possible and now is it possible. Here it's very clear, especially on the right. Now we go to step 3. So that was pretty same to step 2 anyway. 2 biological stains with different concentration. We took all the negative. In this case, we arrived up to almost 97% of efficiency. So this means that this protocols is the right one. It is working. Definitely this is working. More pictures, but always the same. Well, now I have a question for you during my presentation. Do you think that the protocol number 2 works better than protocol number 1 or not? I cordially invite the students to interact now if you would like to, talking with me. Then, of course, at the end of presentation, we'll have time for question, but it will be great to have your opinion about this. Any ideas? No. Okay, no problem. Well, the answer is absolutely not. And you know why? Because it's not the protocols who make the efficiency. It's the color of background. So the combination of protocol number 1 plus protocol number 2 permits us to reach more than 95% of efficiency. Not even the single one, but the combination. Of course, forensic operators have to be trained properly because they have to know the quality of each protocol, and they have to understand and to know which protocol they have to use according to the item under investigation. So it's a very specialized job in this case that you have to do. In fact, the last part of my research time in Copenhagen, it was to training the technicians in the crime unit section, which I used to work, because they must to be able to understand the protocols and they have to know how to choose that properly, which protocols they have to use for a screening of the finding from the crime scene investigation. And now we can see a real case. This was a case in my department, because we were also deputy from the crown prosecutor to the crime scene to the crime investigation. We didn't do crime scene investigation. We just do forensic laboratory analysis there. Well, this jacket was from an homicide case happened in the suburbia of Copenhagen in 2017. Well, I shortly describe it to you. It was a case in which a person, probably for drug dealing or similar things, he was killed by another person with several hits on his head with an hammer, probably. So his head was totally destroyed. Well, a person was arrested just the day after the homicide. This person admitted that he was there when this person was killed, but he also say, I was there, I didn't help, I didn't call the ambulance, but I was not the offender. So he say, I am guilty for a minor crime, because if you don't call the ambulance or the police, it's a crime too. But it's a minor crime, it's not an homicide. So he declared he was guilty for a minor crime. And he declared that the third person was the responsible of this homicide. So the police went to this third person to question him, and they also collected some clothes according to the descriptions of the first person arrested, especially this jacket, because the first person arrested told to the police that the offender used to wear a jacket. This kind of jacket, in fact, was found at his home. But as you can see, we can't see anything at all. We can't see any evidence. In fact, my colleagues tried because the prosecutors asked us for DNA analysis. We did. Well, nothing useful because or we don't find any DNA or we just find mixture DNA. So nothing relevant for this investigation. So the prosecutor asked us for further investigation because it probably was on the direction that this person was really the offender. So he asked us for further investigation. My boss at that time asked me. At the time, I didn't publish. Now this study is published. Before it was not. It was the 2017. I was still doing the research. So he asked, Eugenio, is it theoretically possible to apply this technology for this case investigation or not? I told him, theoretically it's possible, but I cannot guarantee any kind of result if the DNA already showed nothing. They say, okay, it's not a problem. We have the authorization from prosecutors. So let's do this. Okay. I was made in charge of this investigation. I went there with the technician. I start to investigate this jacket. And this is what I found. I found 30, more than 30 different evidence. And especially this one was very relevant in this crime scene investigation because it was a long sign from the chest to the belly area, almost 25 centimeters. And according to what happened to the crime, so Amorites, so what happened? First shot broke the head of the victim, blood will came here. When you are going to give another shot, you'll do this, and blood will do this. So I hypothesize, in fact, I've also found on this part of the jacket some sign which can lead to blood stains. So in my opinion, that one could be an evidence of the victim's blood. Of course, this jacket hasn't been washed before of the collection of the police. This is why we were not able to see anything. So it was also difficult for us to identify stains. I had the opinion that that one was the blood of the victim, potentially. So I documented it. I asked to my colleagues to cut. So to go inside, take outside, because it was a jacket, to analyze inside of the jacket and make two tests. One for blood, human blood, and one for DNA, because we need that information. Well, both tests, blood, yes, positive, DNA from the victims. Thanks to this information, we were able to prove that this person was really the responsible of this homicide. Otherwise, this was probably just still for the city. So it was the first, also not just research, but the proof that this kind of technology is it very important in the crime investigation, in the forensic investigation. This was the first application ever that we did. Now, of course, we are continuing to do this. In my research center in Italy, we are working with this technology. So we use this technology now every day for crime scene, for laboratory investigation, etc. Well, I had also to do some more thing when I was still there. Just a quick description of how I tried to improve the quality of photo documentation. Why? Because photo for me is very important. It's my electropherogram. a good picture is my electrophysical. So I need to improve as much as possible this field. Before we started with the manual, I had a technician with me and he manually made the photo. Then we start to use um, a new system, totally IT based, in which a camera was standardized and I had the possibility to control the camera just with a computer. And you can see how is better now. You can see the comparison of the two pictures. So that was great. Now we are working like this, actually. And I also was interested to see if it was possible to improve the dilution and still have the possibility to identify some biological states. I was going to calculate the minimum quantity of stain still identifiable through these methods of analysis. So minimum quantity detectable, it was just a new parameters. What I did, I used the same approach of before, negative control, positive control, just I put in the middle dilution 116, 132, 165. Please note that dilution 165 means that we are adding less than 1 microliters of cells. And this means very, very small quantity of the DNA, which is probably in picogram now. Okay, because we have to do the conversation. Well, in some case, only in some cases, for example, in jeans and in white shirt, we were able to identify some stains. And that's great. That's a great result because this is the proof that we are able to identify even the smallest stains. This means for the justice the possibility to get lots of information, of scientific information for it. Conclusion, of course, the detection of the biological stains is affected anyway from the color of the background of the item. It's easy to see on white than on black and dark shirt. Each fabric has a specific minimum quantity detectable parameters, and we are able to get the best result in fabric like the jeans or the white shirt. Well, these are the future perspectives in which I finished my Copenhagen research experience and I continue. Nowadays, we are still working on this one. For example, development of an ALS spectrum for the identification of biological tissue, comparison between existing and new tests, but also specify much better about these new parameters that we found out. And also, is it important for the cold case investigation, the evaluation of the time effects on the stains and on the alternative light source investigation. Well, that's all from my side. Thanks for your attention, and I'm here for get your questions.
Thank you, sir, for the engaging and profound session. If the students have any question, they can unmute and ask the question one by one. You also have a provision to type the question in the chat box.
Yes.
So during your presentation, there were threads of questions, so I'll just read them out. The first question is, what precautions and consideration should forensic professionals take when using alternative light source to avoid contamination or misleading results in their investigation?
Yeah, first of all, actually, we are also working on specialist procedures as per the DNA procedures and analysis. First risk in our laboratory is the contamination. So we think that DNA analysis and biological identification must happen in two separate places. Okay, two separate rooms, at least two, but maybe more than two, of course, because we have to separate totally for prevent any kind of contamination the area in which arrives the items, in which we open the items from the security bag, and in which we perform the screening with this kind of biotechnologies. When we collect, only the sample collected or the piece of the fabric that we cutted have to go in the other rooms in which the DNA analysis will take place. The rest have to go back. Never put some finding or some security bag with finding from crime scene, so totally contaminated, in a further room. Otherwise, we'll just risk to insert a mistake into the system. And if we do this, everything will collapse. And we are not able to provide justice with scientific data, because how can we know about the correctness of our data or about the contamination or whatever else? This is why actually we are also working a lot on the quality standards on our lab. For example, the research center which I now am in charge, which I'm the director, we are just specialized on this investigation. So our lab have an external room in which we do the reception, the acceptance of the findings that the police give us. We sign the reports. Then it's only me and my colleagues that go inside the room of analysis, and we follow a very specific way in different sub-area, in which for each sub-area there is a technical operation. And everything will finish at the last sub-area, in which we will have the samples collected. We put again in a security bag, in a new security bag. Everything must be documented. The chain of custody must be guaranteed in this case. And then only this sample we will send to another laboratory for do the genetic analysis. And the fabrics, we will call again the police, and they will come to my lab for getting back the rest of the findings after our analysis. This is the procedure that actually we are using here in Italy.
So there is another question. Are there any advancement or recent innovation in alternative light source technology that have improved forensic analysis and investigation processes, specifically in Italy?
Well, it's not this target of research is not a very famous one, because there is a lot of attention on DNA, probably too much. DNA is very important. It's absolutely needed and is a new frontier of the analysis, but it's not anything. For example, if we don't use this technology as properly, we lost DNA. So we have to know that we are in a group. We must be in a group. So I invite the students, the nowadays students, when they will do their thesis or they want to apply for PhD or whatever, I invite them to explore new ways of the research. Don't just get stuck in which is already open, because yeah, they are important and they are the future too. But this is also very important things related nearby. DNA, it's important, but it's not everything in forensics. We have to keep this very clear. This is a way that we can assist. It's a little bit innovative also as what we projected our lab, etc. From the Italian situation, well, there is also my private center who is accredited with the Ministry of Justice. There are other two labs, one from Italian police and another from Italian Carabinieri, which are the military police, that they are accredited, but they are accredited for DNA analysis. They have not the accreditation for this biological investigation previously. Of course, they can do, and they do every day. but sometimes the procedures are pretty critical. So when I have to when I work as a consultant for Ministry of Justice or for Defense or whatever else, and I read in a technical report the methods used by these colleagues, and I see critical stuff, I will highlight to the court. This is also the work of the forensic scientist. We can do both laboratory analysis, but also technical review for the court or for the lawyers. This is very important too. So, students, it's very large, the field of professional application for you. You have to take both in consideration, not only the laboratory experience. That's important to do laboratory experience, but the profession of the forensic science doesn't stop in a lab. We also are very active in court. I also work for the crime scene investigation in some cases here in Italy because I got some certification, etc. So the application of your degree is quite large. So be open-minded for your future career.
So this is going to be the last concluding question that we'll ask, though it's a little deviation from our topic of today, but I'll still ask since it's been put up by one of the students. What role do alternative light sources play in detecting and visualizing bruising and other injuries on a victim's body during forensic examination? So if you can comment.
During bruising, can you please repeat bruising, during bruising?
What role do alternative light sources play in detecting and visualizing bruising and other injuries on victim's body during forensic examination?
Okay, so this is more a medical legal question. So it will be correct that you will ask to a medical legal this question. I'm a forensic biology, but from a theoretical application, I can confirm that is it possible to use this kind of technology also for evaluate the injuries on victim's body, if alive or not alive, no matter, you can use. For example, I don't do because I'm not a fingerprint expert, but theoretically, we can use the alternative light source also for fingerprint investigation. So the application of this technology is very large. I am a geneticist, a biologist. So this is why I told you about the biological effects of this technology. But there is not the only one, of course. So about this, there is a large way of application. Good question.
Thank you, sir, for patiently answering the student's question. Gratitude is a fairest blossom that springs from the soul. On this note, I request Umair Razak, third semester MSc student, to deliver the vote of thanks.
I would like to extend my heartfelt gratitude to our esteemed guest speaker, Dr. Eugenio D. Orio, for gracing us with his insightful lecture today. Sir, your presence and words have truly enriched our understanding and broadened our perspectives on the application of alternative light source to reveal hidden truths in forensic investigation, unveiling evidence beyond the visible spectrum, and shedding light on the unseen, crucial details that aid justice. Sir, your willingness to share your expertise with us is truly commendable, and we are grateful for the time and effort you have invested in preparing and delivering this lecture. Once again, thank you, sir, for your enlightening lecture. We look forward to the possibility of welcoming you back in the future. Your presence has made a significant impact, and we are truly grateful. This session would not be possible without the support and guidance of the members of the management led by the principal, Reverend Father Dr. Augustine George. Thank you, fathers. I extend my gratitude to Dr. Callistus Jude and Professor Don Cairo for encouraging us to strive better and helping us conduct such programs. Thank you, sirs. The smooth conduct of the guest lecture is because of the faculty members of the Department of Forensic Science. I thank all the dear teachers for coordinating the program to make it a success. And a special thanks to Mr. Mayank Raibode for ensuring a technically smooth session. I also thank all the students of MSc Forensic Science for being a great audience. I request the participants to turn on their videos for a photo screenshot of the session. Thank you one and all. The feedback form link will be shared with the participants. Kindly fill it out without fail.
Thank you, Professor Eugenio, for that lecture. Now I'll request Professor Don Cairo to.
Thank you so much, sir, for taking time and for being with us today. Definitely, the students will be benefiting from your words and your experience that you have shared with them. And we're eager to have further interactions with you. And if everything goes well, then we can also have you down physically, with us, which will definitely be a boost to our students.
Thank you so much, Professor. I told you it was a great pleasure for me to interact with you, with your department. Many thanks again to Dr. Sushita, who just got in touch with me, and to all your department. And I really appreciate time and also the active participation of your student delegation. It will be a pleasure to have a future initiative all together. It will be a really pleasure of mine.
Thank you.
Thank you
So much. Thank you so much. Thank you.
If there are any more questions, maybe also in future, you can just have my mail. You can just keep in touch, you or the students, whatever. It will be just a pleasure.
Definitely. Thank you so much, sir. Thank you.
Okay. Thank you so much.
I would request the host to end the meeting.
Okay. Thank you. Have a good evening.
You too.
Bye.